Have you ever really stopped to think about what's happening beneath your feet? You know, anchoring every plant you see, from like a huge tree down to the smallest blade of grass.
It's this whole hidden world, isn't it? An incredible feat of biological engineering, really.
Absolutely. Welcome to the deep dive. Today, we're digging into the fascinating world of plant roots. We're using a chapter from the Raven Biology of Plants textbook as our guide.
Our mission today is pretty straightforward. Unpack the core functions, the different structures, and the really amazing growth processes that make roots the unsung heroes of the plant kingdom.
Yeah, and we want to make it clear and understandable. We'll
Mm.
break down the complex terms so you walk away feeling like you really get it without getting bogged down in, excessive detail. We'll cover everything from how they first pop out of a seed to some really specialized adaptations.
And understanding roots, it's not just for botany majors. It's actually key to appreciating how plants survive, how they thrive, and even how they shape entire ecosystems.
Couldn't agree more. So let's dig in. Where do we start? What are the absolute essential jobs that roots perform?
Okay, so at the very core, you've got four main jobs. First, anchorage. The root is literally the first thing out of a germinating seed, holding that seedling tight.
Right, you got to stay put.
Exactly. Second, absorption. They're constantly pulling in water and essential minerals from the soil. Can't live without those.
Makes sense. What else?
Third, storage. Think about carrots, sugar beets, sweet potatoes. Those are roots acting like pantries, storing food made up in the leaves.
Ah, okay. So food made upstairs gets sent downstairs for later.
Precisely. The phloem carries it down. Then, fourth, conduction. They transport that water and minerals up through the xylem, and they can also transport stored food back up when the plant needs it, again using the phloem.
So anchorage, absorption, storage, conduction. The big four. But you mentioned they do even more.
Oh yeah, much more. They're little chemical factories down there. They synthesize hormones like cytokinins and gibberellins, which travel upwards to stimulate growth in the shoots.
Really? I thought hormones were mostly made in the shoots.
Some are, but roots make crucial ones too. They also produce secondary metabolites, nicotine and tobacco, for example,
Yeah.
made in the roots, then shipped up to the leaves.
Wow, okay. That's surprising.
It is. Plus, they can be involved in clonal regeneration, basically, new shoots sprouting directly from a root. And they actively manage their surroundings, redistributing water in the soil, secreting compounds called exudates.
Exudates, what are those?
They're substances the root releases into the rhizosphere, that's the soil zone immediately around the root. It influences the microbes there, nutrient availability, all sorts of things. It's a very active interface.
Okay, so roots are way more dynamic than just passive straws in the ground. What about the overall structure? Are all root systems built the same?
Not at all. There are two main blueprints, basically. Most seed plants, except for the monocots, develop what's called a taproot system.
A taproot, like a carrot.
Exactly like a carrot. The primary root, the first one from the seed, grows strongly downwards. That's the taproot. And then smaller lateral roots branch off from it. Older ones near the top, younger ones near the tip. They tend to go deep.
Okay, so that's one type. What's the other?
The other is the fibrous root system, which you see in monocots. Think grasses, corn, oats. In these plants, that initial primary root doesn't live long.
Oh, so what takes over?
The main system develops from adventitious roots. These actually arise from the stem near the base. These roots and their branches form this dense, shallow mat. There's no single dominant root.
Ah, so like a net just under the surface.
Pretty much. It's fantastic for holding onto soil and preventing erosion, which is why grasses are so good at it.
Yeah.
But they don't usually penetrate as deeply as taproots.
That makes sense. Different strategies for different plants. How responsive are roots to their environment? Do they just grow randomly?
Oh, far from random. Roots are incredibly sensitive. They respond to gravity, light or lack thereof, moisture gradients, temperature, nutrient patches.
So they can sense where the good stuff is.
Absolutely. They show amazing developmental plasticity. If part of the root system encounters a pocket of soil rich in, say, nitrogen or phosphate, it will quickly ramp up the development of lateral roots right into that zone to maximize uptake. It's very targeted.
That's clever. And
Yeah,
the
sure.
sheer size of these systems can be unbelievable, right?
That's mind-blowing sometimes. You hear about trees like Boscia albatrinca in the Kalahari with roots found 68 meters down.
68 meters. That's insane.
Right. Mesquite roots have been found over 50 meters deep. Even alfalfa, a forage crop, can send roots down 6 meters. And it's not just depth. The lateral spread can be huge, often 4 to 7 times wider than the tree's crown.
Wow. So the part we see is just the tip of the iceberg. literally.
Definitely. Though it's important to remember that most of the really active absorption happens relatively close to the surface. The majority of the fine feeder roots are typically in the top 15 centimeters or so of soil.
And those are often the ones with mycorrhizal fungi.
Exactly. Those fungal partnerships dramatically extend the plant's reach for water and nutrients way beyond what even the root hairs can do alone.
Speaking of root hairs, there's that famous study on the rye plant.
Ah, yes, the rye plant study. It's a classic. They looked at a single rye plant, just 4 months old, grown in a relatively small amount of soil.
And the numbers were staggering.
Unbelievable. The total surface area of its roots was estimated at 639 square meters. That's 130 times the surface area of its leaves and stem.
Yes, wow.
And get this, it had an estimated 14 billion root hairs. If you laid them end to end, they'd stretch over 10,000 kilometers. That's the kind of absorptive surface we're talking about, mostly unseen.
It really highlights this idea of balance, doesn't it, between the roots and the shoots.
It absolutely does. That's the root-shoot ratio. There has to be a functional balance between the photosynthetic area above ground and the absorptive area below ground.
And this ratio changes over time.
Yeah, typically seedlings have a very high root-shoot ratio, lots of root relative to the shoot. As the plant gets bigger and establishes, that ratio usually decreases a bit.
So if you damage one system, the other feels it.
Directly.
Yeah.
If the roots are damaged, say, by drought or disease, the shoot growth slows down because it's not getting enough water, minerals, or those root-produced hormones.
And the reverse.
If you heavily prune the shoot, root growth can be limited because the roots aren't getting enough carbohydrates from photosynthesis or shoot-produced hormones. It's a tightly linked system.
And those fine feeder roots, they're not permanent structures.
Not at all. They're very dynamic. Fine roots are constantly dying off and being replaced. It's estimated that maybe a third of the total energy captured by land plants each year goes into just producing and replacing these fine roots. It's a huge investment.
That explains why transplanting can be so tricky. You inevitably lose a lot of those fine roots.
Exactly. And that's why gardeners sometimes cut back the top of a plant when they transplant it. They're trying to rebalance that root-shoot ratio to compensate for the root loss. Same idea when repotting a root-bound plant, you give the roots more space to restore the balance.
Okay, let's shift focus to how roots actually grow at the tip. It's not just random elongation, is it?
No, it's highly organized. Root growth is usually pretty continuous unless conditions are bad, like drought or cold. And the very tip is protected by this structure called the root cap.
Right, you mentioned that. Like a little hard hat?
Sort of, yeah. It's a thimble-shaped cap of living cells covering the very end. Its main job is to protect the apical meristem right behind it. That's the zone of active cell division, the ultimate source of all the root's cells. It's very delicate.
How does it help the root push through soil? That seems like it would cause a lot of friction.
It does, but the outer cells of the root cap secrete loads of mucilage, a slimy polysaccharide substance. It acts like a lubricant.
Ah, clever. And what about those border cells you mentioned earlier?
Right, those are fascinating. The outermost root cap cells are actually programmed to separate from the cap and from each other as the root grows. They become these individual living border cells that hang out in the rhizosphere.
And they're not just dead debris.
Not at all. They stay alive for weeks, change their gene expression, pump out specific proteins. They're thought to do a lot. Protect the meristem from pathogens, help maintain root soil contact, mobilize nutrients, maybe even act like tiny ball bearings to reduce friction.
Wow. So the root cap is more than just a physical shield.
Much more. It's also a sensory hub. Inside the cap, there's a central column of cells, the columella. These cells contain starch grains that settle according to gravity, allowing the root to perceive down.
Gravitropism.
Exactly. The columella helps detect gravity, and it also seems involved in sensing water potential gradients, guiding the root towards moisture hydropism. So the root cap is constantly sensing and signaling back to the growing regions to direct growth.
Okay, so the cap protects and senses. Where does the actual increase in length happen?
Right behind the root cap and the apical meristem is the region of cell division. That's where the new cells are produced. But the main increase in length happens just behind that, in the region of elongation.
How long is that region?
Usually just a few millimeters. But in that short zone, the newly divided cells expand dramatically, pushing the root tip forward. Crucially, above this region, the root cells don't elongate anymore. The root doesn't get longer further back.
So only that tiny tip section is actually moving through new soil.
Precisely. And then above the region of elongation, you have the region of maturation or differentiation. This is where the cells finish developing into their specialized types, and importantly, where root hairs form.
Why do they form there and not lower down?
Because if they formed in the region of elongation, they'd just get ripped off as that section pushes through the soil. They need to form on cells that are no longer elongating, cells that are anchored relative to the soil.
Makes perfect sense. It's all timed perfectly.
It has to be. And these regions aren't super sharply defined, there's overlap. For example, the first bits of the vascular tissue, the phloem, often mature closer to the tip than the xylem or the root hairs.
Okay, let's zoom in on the internal structure in that mature region. What tissues do we see if we take a cross-section?
Well, root structure is generally simpler than the stem structure, mainly because there are no leaves or nodes. You basically have three tissue systems arranged concentrically. Outermost is the epidermis.
The skin.
Essentially, yes. It's typically a single layer of cells, closely packed. Importantly for absorption, these cells usually lack a thick, waxy cuticle, and they're the cells that produce those root hairs we talked about.
Billions of them in that rye plant.
Exactly. Those hairs massively increase the surface area. They are, however, quite delicate and short-lived, constantly being replaced as the root grows forward from the tip. That's another reason careful transplanting is vital.
Okay, epidermis on the outside. What's next?
Inside the epidermis is the cortex. This is usually the thickest part of a young root, made up mostly of parenchyma cells, the sort of general-purpose ground tissue.
What does the cortex do?
A major role is storage, often starch. It also has lots of intercellular air spaces, which are vital for aeration, allowing gases to diffuse to the inner tissues. In aquatic plants, these spaces can be huge, forming a tissue called parenchyma.
How do water and minerals move through this big cortical area?
Two main ways. Either through the interconnected cytoplasm of the cells, moving cell to cell via tiny bridges called plasmodesmata, that's the symplastic pathway, or they can move along the interconnected cell walls and intercellular spaces, that's the apoplastic pathway.
Okay, simplest to the cells, apoplast around them. Is there anything controlling that movement?
Yes, absolutely. The innermost layer of the cortex is the endodermis. This is a critical control point.
How so?
The endodermal cells fit together tightly, no gaps. And crucially, their radial and transverse walls, the ones perpendicular to the surface, have this special band embedded in them called the Casparian strip.
Casparian strip. What's it made of?
It's impregnated with suberin, a waxy, waterproof substance, and sometimes lignin too. Think of it like mortar between bricks, but waterproof mortar.
So it blocks something.
It blocks the apoplastic pathway. Water and solutes moving along the cell walls hit this waterproof barrier and can't go any further around the endodermal cells.
Ah, so they're forced to go through the endodermal cells.
Precisely. They have to cross the cell membrane of the endodermal cell to get into the vascular tissue. This means the living membrane gets to control what passes. It's a selective barrier, ensuring the plant only takes up what it needs and keeps out potentially harmful stuff.
That's incredibly elegant. A selective checkpoint.
It really is. And in many plants, there's often an exodermis just under the epidermis, another layer with Casparian strips, providing an additional barrier and reducing water loss from the cortex back to the soil.
Okay, so epidermis, cortex with its endodermis checkpoint.
Right.
What's at the very center?
That's the vascular cylinder, sometimes called the stele. This contains the plumbing, the primary xylem, and primary phloem, and it's all enclosed by one or more layers of non-vascular cells called the pericycle.
Pericycle, what does that do?
The pericycle is really important. Firstly, it's the tissue where lateral roots originate in most seed plants. They actually start growing from the pericycle outwards. Secondly, in roots that undergo secondary growth, the pericycle contributes to forming the vascular cambium and the first cork cambium.
So it's a source of new tissues. How are the xylem and phloem arranged inside?
Typically, the primary xylem forms a central core, often star-shaped or ridged in cross-section. The number of arms or ridges varies, could be 2, 3, 4, 5, or many, like in maize. The phloem strands are located in the valleys between these xylem ridges, right up against the pericycle.
Okay, I can picture that. Xylem in the middle, like spokes, phloem tucked between them. Now, you mentioned secondary growth. What happens when a root starts getting woody?
Right. Secondary growth increases the root's diameter. It involves two lateral meristems, the vascular cambium and the cork cambium. Remember, monocots generally don't do this.
So how does the vascular cambium form?
It starts from undifferentiated precambium cells between the primary xylem and phloem, and also from divisions in the pericycle cells located outside the tips of the xylem ridges. These dividing cells link up to form a complete ring, initially wavy, following the contours of the primary xylem.
And then this ring starts producing new tissues.
Exactly. The vascular cambium divides to produce secondary xylem wood towards the inside and secondary phloem towards the outside. Because it produces more xylem than phloem and starts producing cells faster on the inside curves, the cambium quickly becomes circular.
What happens to the original primary tissues?
The accumulating secondary xylem pushes everything outwards. The primary phloem gets crushed and often obliterated, though sometimes tough phloem fibers might remain. The primary xylem stays right in the center, surrounded by the secondary xylem.
So the root is getting thicker. What protects this growing root? The epidermis can't stretch that much.
It can't. As secondary growth proceeds, a cork cambium arises, usually from the pericycle. This cork cambium produces cork cells, phloem, to the outside. These are dead, waterproof cells and sometimes a layer of living parenchyma called pheloderm to the inside.
And together these form.
Together, the cork cambium, the cork, and the pheloderm make up the periderm. This is essentially the bark of the root, replacing the epidermis and cortex, which get sloughed off.
Does this periderm allow for breathing?
Yes, the periderm develops lenticels, which are porous areas with intercellular spaces allowing gas exchange between the internal root tissues and the soil environment. So a mature woody root has a central core of primary xylem, surrounded by secondary xylem, the vascular cambium, secondary phloem, maybe some primary phloem remnants, the pericycle, and finally the protective periderm.
That's quite a transformation. You mentioned lateral roots originate from the pericycle. Can you walk through that process?
Sure. It's a key feature. Lateral roots are endogenous, meaning they start from inside the parent root, specifically the pericycle, usually opposite a protoxylem pole, one of the xylem ridges.
Okay, so deep inside, how do they get out?
Cells in the pericycle start dividing, forming a little bulge, the lateral root primordium. This clump of cells organizes itself, forming its own apical meristem and root gap very early on. Then it literally pushes its way outwards through the cortex and epidermis.
Does it digest the cells in its path?
It's thought that it might secrete enzymes to help break down the cortical cells, basically carving a path to the outside. Once it emerges, its vascular tissues need to connect up with the main vascular cylinder of the parent root, which happens as the cells between them differentiate.
Fascinating. Roots aren't just standard anchorage and absorption units, though. There are some really cool modifications, right?
Oh, definitely. Plants have adapted roots for all sorts of specialized functions. Think about aerial roots, roots growing from stems or leaves up in the air.
Like on orchids.
Orchids are one example, yes, but also things like prop roots on maize or mangroves. They grow down from the stem to provide extra support. Banyan trees have incredible stilt roots coming down from branches, and things like ivy have clinging roots that help them climb walls.
What about roots that help plants breathe in waterlogged soil?
Ah, yes, pneumatophores. You see these in plants like mangroves growing in swamps where the soil is anaerobic, lacking oxygen.
How do they work?
They're amazing. They grow upwards, against gravity, sticking out of the mud or water like little snorkels. They're covered in lenticels for gas exchange and have spongy internal tissue, arenchyma, to conduct air down to the submerged parts of the root system.
Like built-in breathing tubes. And the orchid roots you mentioned.
Many epiphytic orchids, those that grow on other plants, have roots covered by a unique multi-layered epidermis called the velamen.
Velamen.
Yes. It's spongy, often white or silvery. It helps absorb atmospheric moisture and nutrients, reduces water loss from the root core, provides physical protection, and in some cases, it even contains chlorophyll and photosynthesizes.
Wow, photosynthetic roots. And then, of course, there are the roots specialized purely for storage.
Right, the fleshy roots we eat. Carrots, sweet potatoes, sugar beets, radishes. Their primary modification is having a huge amount of parenchyma tissue for storing carbohydrates or water.
Is the storage tissue always in the same place?
Not exactly. In a carrot, most of the fleshiness comes from abundant parenchyma in both the secondary xylem and secondary phloem produced by the main vascular cambium.
Okay. What about sweet potato? That's a root, right?
Yes, it's a modified lateral root. Its development is more complex. Extravascular cambium develops within the secondary xylem, around the vessels, and this additional cambium mostly produces lots and lots of storage parenchyma cells, filling up the root.
And sugar beets. They get huge.
Sugar beets are different again. They form the regular vascular cambium, but then additional concentric cambia form outside the previous ones. Each of these supernumerary cambia produces secondary xylem and phloem, but Again, it's heavily dominated by storage parenchyma. That's how they bulk up so much. It's also worth noting that the very top part of some fleshy roots, like beets or carrots, is often derived from the hypocautal, the embryonic axis below the cotyledons, technically part of the stem.
So much variation. How does scientists figure all this intricate development out? I know Arabidopsis thaliana is a big model
It
organism.
is. Arabidopsis, that little mustard weed, is fantastic for root research. Its root is relatively simple. You can grow seedlings easily on agar plates where the roots grow straight down, making it easy to see defects. And it's incredibly easy to manipulate genetically. We know its whole genome sequence.
What has Arabidopsis taught us about how root cells know what to become?
A lot about pattern formation. The Arabidopsis root has a very precise radial pattern: epidermis, cortex, endodermis, pericycle, vascular tissue. Even within the epidermis, cells destined to become root hairs form in specific locations relative to the underlying cortical cells.
So it's not random.
Not at all. There are hair cells, H position, and hairless cells, N position. Studying mutants helped figure this out. For instance, mutants lacking a functional TTG gene develop root hairs on all epidermal cells, regardless of position.
So TTD controls the pattern, not hair formation itself.
Exactly. It showed that positional information is crucial. This led to some really elegant experiments using lasers to zap specific cells in the root tip.
What did they find when they did that?
They found something revolutionary. If they ablated, say, the 4 cells of the quiescent center, the very inactive cells at the heart of the meristem neighboring cells from the vascular cylinder would actually move in, divide, and take on the fate of quiescent center cells, even though their lineage was different.
Whoa. So cells can change their destiny based on where they end up.
That's the key insight. It showed that cell fate in the Arabidopsis root seems to be determined more by positional cue signals from neighboring cells, their location within the overall structure, rather than strictly by their cell lineage, meaning who their parent cell was.
So it's less about predetermined fate and more about responding to the local environment.
Precisely. It suggests the apical meristem isn't just an autonomous machine churning out cells according to a fixed plan, but that it's constantly receiving feedback and positional information from the cells it produces. It's a much more dynamic view of development.
Okay, that's a lot to take in. Let's try to summarize the key takeaways from our deep dive today.
All right. First, I'd say, remember, roots are incredibly multifaceted. Not just anchors, but vital for absorption, storage, transport, hormone synthesis, defense. And they're remarkably adaptive.
And that diversity in design is striking. Deep taproots, shallow fibrous systems, aerial roots, pneumatophores, each structure fits a specific ecological niche. Form follows function.
Absolutely. Then there's the dynamic growth process. That root tip is a highly organized engine. The protective root cap, the distinct zones for division, elongation, maturation, all working together for continuous directed growth.
And the internal structure shows incredible precision, especially that endodermis with the Casparian strip acting as a selective gatekeeper, controlling exactly what gets into the plant's vascular system.
Don't forget secondary growth either. For woody plants, it's a massive transformation, adding strength and replacing the primary outer tissues with protective periderm. It's ongoing adaptation.
And finally, the cutting-edge research, like with Arabidopsis, shows us development is often about positional information, not just fixed lineage. Cells are remarkably flexible in determining their fate based on their neighbors.
It really highlights the complexity and elegance hidden beneath the surface.
So the next time you're out and you see a plant, maybe take a moment to think about that incredible hidden world beneath the soil. That silent, complex, dynamic engine constantly working away. It makes you wonder, what other hidden biological processes are happening all around us, just waiting for a deep dive?
It definitely gives you a new appreciation for the complexity of life, even in something as seemingly simple as a root.
Well, thank you for joining us on this exploration of plant roots. We hope this journey into the plant's unseen foundation has been illuminating. From the entire Deep Dive team, thanks for listening.